Hi, I'm Carla Perpiñá-Clérigues, and I'm going to present you the following poster entitled Alcohol Consumption Affects the Lipidome of Extracellular Vesicles, a sex-based difference study. The neuroinflammatory effects of alcohol can be irreversible, leading to neurostructural and behavioral alteration. So it's important to study the mechanisms involved in the neuropathology of alcohol. It is known that lipid composition of extracellular vesicles is involved in the formation, release, and function. For example, lipids participate in the stability of the EVs in the cellular environment and interaction with cells. Significantly, lipid dysregulation has been associated with neurological and inflammatory issues. In this manner, exosome lipidomics is emerging as a novel field for finding novel lipid species with biomedical application. On the other hand, prior studies have also shown sex-based differences in the microRNA profiles of EVs isolated from adolescents after binge drinking. Thus, due to the role of EVs lipids and differences in other vesicle components resulting from alcohol consumption and sex, the main aim of this study is to investigate how ethanol alters plasma EVs lipids composition differently depending on sex and involvement of the immune response. For that, the following methodology was used. Exosomes were isolated and lipids were extracted for quantification and identification by liquid chromatography mass spectrometry. Then, after an exploratory analysis, the experimental groups, ethanol and control groups of females and males, were compared by differential abundance analysis. To assess biological function, lipids were annotated into three hierarchical levels: superclass, main class, and subclass, using the databases REMMED and lipid maps, followed by a class enrichment. This strategy was carried out separately in adolescents after acute ethanol binge drinking and in adults after chronic drinking. The comparisons in the differential abundance lipid analysis and lipid set enrichment analysis were three. Comparison between alcohol females and control females, the same in males, and the comparison of these two comparisons. As a result of differential abundance analysis, in both cases, adolescents and adults, we observed lipids that display significant changes in abundance in ethanol intoxicated females and males compared to respective controls. Some are sex-specific, and others are shared, although the behavior may be opposite between sexes. For instance, in this case. On the other hand, the results of the lipid set enrichment analysis in adolescents indicate that ethanol induces a differential enrichment of extracellular vesicles lipid classes in human female adolescents when compared to males. These lipids participate in the EVs formation, release, and uptake, in inflammatory immune response, and the TLR4 activation, suggesting that binge alcohol drinking in female adolescents could be associated with higher extracellular vesicles biogenesis and a more significant spread of neuroinflammatory than in males. Also, in adults, we see certain enriched classes in the differential experimental groups. At the moment, we are investigating biological functions. In conclusion, this study supports the existence of differences between female and male subjects in the extracellular vesicles lipidome during the response to alcohol consumption. Also, function of these lipid species suggests that the binge alcohol drinking in female adolescents could be associated with higher extracellular vesicles biogenesis and a more significant spread of neuroinflammation than in males. Therefore, this work takes a novel approach to study the sex-based differences in the effect of alcohol based on bioinformatic analysis of lipidomics data, and suggests the use of lipid sex specifics in extracellular vesicles as a suitable candidate for non-invasive biomarkers that could help to explain the mechanisms underlying the neuroinflammatory response after acute intoxication in adolescents and chronic drinking in adults. That is all, and thank you for listening.